So, this third part of chapter 12 is really going to look at the mechanism behind DNA replication. So, we're going to take you step by step through every single process of DNA replication. And a lot of those different processes really have to do with different substrates. So, this is a really good table for you guys to get memorized. The components that are involved in DNA replication, if you know what they are and you know what their function is, then the process becomes very easy to memorize and to work through. So we're just going to start with our initiator proteins, which is the beginning of the DNA replication process. So keep in mind at this point in time, if we are in a bacterial system, then we've got one chromosome that is double-stranded DNA, and that system can go through either theta replication or rolling circle replication. If you think of theta replication, where you have bidirectional replication for the next few slides, that might make comprehending this a little bit easier. In addition to theta replication, linear eukaryotic replication is where we have multiple sets of chromosomes that are contained inside the nucleus that are going to be synthesizing themselves during the S phase of the cell cycle. So that's really where we're at. So when it comes to starting the process of copying the entire genome, the first set of proteins that are involved with this process are initiator proteins. So very simple, they initiate the DNA replication process. So initiator proteins are going to bind to the replication origins and start causing DNA to begin unwinding. And we can see over here in this figure that our initiator proteins are going to be kind of globbed up all together. What they end up doing is attracting the DNA material, the chromosome. It flips it around in like almost a figure 8, and that is the signal for DNA helicase to come in and start unwinding, as we can see in step 2 here. So initiator proteins have to bind to the origins of replication, which cause DNA to unwind, and then helicase can come in and go to work. So helicase is its main job is to unwind DNA. So DNA helicase here is going to be this kind of darker yellow protein that is actually setting on either the leading or the lagging strand of DNA, and what it does is it moves along the double-stranded DNA and clips the hydrogen bonds that are holding one strand to the other. So after DNA helicase has come in and the replication bubble has begun to form, then we have single strands of DNA that aren't connected to one another. So DNA is really unstable at this point. So in order to make it stable, what ends up happening is that the single-stranded binding proteins, these little purple ones, will actually come in. You can see them right here, and they are going to orient themselves in a pair or in a set of 4 around the single-stranded DNA, and that's going to help to stabilize DNA so that that replication bubble stays open for the replication process to take place. So these single-stranded binding proteins are going to be all along the anywhere that you see single-stranded DNA occurring. All right, and then DNA gyrase or gyrase, everybody pronounces it differently, is actually a topoisomerase, and this is an enzyme that, much like helicase, and we know that they're enzymes because there's an ASE at the end of their name, and DNA gyrase is going to control the supercoiling of DNA. So if you guys have ever taken a piece of like tie-down rope, the black and orange rope, and tried to unravel it, what ends up happening is as you unravel it into separate threads of the bigger rope, the bottom of the rope actually curls up on itself and gets caught. A lot of it you can also see this like untangling hoses and that type of thing. So that the same concept happens with DNA. So DNA gyrase actually is a protein that will set down around the DNA ahead of the replication fork on either side and actually help to absorb all that torque and smooth out the DNA so it doesn't get really stranded up around itself. So that's DNA gyrase's function. And then the primers are going to be synthesized and put in place by primase. So a primer is all of our DNA polymerases. So that we're going to be dealing with, and there's 5 of them total. 2 of them are really super important, and I'll tell you about those when we get to polymerase. But DNA polymerases are going to require a 3' OH end. Therefore, and they require that so that they know where to come in and set down on the DNA strand to start the synthesis process. So what ends up happening is that the primase enzyme is actually made up of RNA, not DNA, which means that it has the uracil nucleotide instead of thymine. And that little chunk of RNA that we call a primer that is made from primase will be synthesized, and it will actually come in and be bound to the origin of replication. And that origin of replication is then going to be the signal point to have DNA polymerase come in. It's going to be the target that DNA polymerase is going to be looking for. So again, our primers are made by primase, and it's in the form of an enzyme that's actually going to bind the primer to the origin of replication so that DNA polymerase can find its starting site. All right, so let's get into talking about DNA polymerase a little bit. Realize that this is a really big, important enzyme. We know that it's an enzyme because it has ASE at the end of it, but there are about 5 different forms of DNA polymerase. The one that we're going to talk about first is DNA polymerase 3. And even though it's not DNA polymerase 1, it's really not conducive to teaching, but DNA polymerase 3 was actually the polymerase that was discovered third. So it got the 3 designation, but it happens first. So it is really important in the replication process because it is going to be utilized first. So DNA polymerase 3 is going to be a really big enzyme that is going to come in and attach to available 3' OH groups. And this is going to be on the primer. And then DNA polymerase 3 is going to polymerize the new stretch of DNA. Okay, and polymerize is a fancy word for create. So it's going to bring polymers in together, start binding them to the 3' OH end, and start synthesizing that new strand of DNA based on the roadmap provided by the DNA template or the old strand of DNA. So on this figure here, let's just make sure that we can orient ourselves. We've got DNA synthesis happening. Our gray DNA is the template strands. We have a leading strand and a lagging strand, and then the newly synthesized DNA is in red. And the green part to DNA, that is the primer. So that's going to contain RNA material or uracil, and that is going to be what DNA polymerase 3 is looking for so that it can set down across the template strand and begin synthesizing that new strand of DNA. All right, so just another look at it. And keep in mind as we talk about the direction of replication, we had the leading strand and the lagging strands. So we can get continuous replication occurring on the leading strand, but eventually we're going to have to stop and create another lagging strand because those replication bubbles are going to get bigger and bigger and bigger as we synthesize more of the genome. And then on the lagging strand, you can see down here that is associated with discontinuous synthesis, and they form Ozaki fragments, and those Ozaki fragments are going to later be glued back together. All right, so this is what this process is really starting to look like in a step-by-step fashion. If we start up here at the top, we've got our DNA gyrase that is actually relieving the tension downstream. We've got helicase that is clipping the hydrogen bonds between the double-stranded DNA, allowing them to become single-stranded DNA. That single strand is going to be stabilized by single-strand binding proteins. And then we have primase that is going to come in and synthesize a short chunk of RNA material as the starting point for the newly synthesized DNA strand. So that is what is DNA primase is actually, or DNA polymerase is actually looking for. So we can see that as we start from the beginning of this process, our replication fork is getting bigger. Our newly synthesized strands are going to get bigger. Here on this leading strand, replication is continuous, and a primer is required only at this 5' end of the newly synthesized strand. Okay, but on the lagging strand that we see here down at the bottom, replication is discontinuous, meaning that we get Ozaki fragments or chunks of DNA being synthesized. And a new primer actually has to be generated at the beginning of every Ozaki fragment. So talking a little bit more in depth about DNA polymerase, we've got 5 main types. DNA polymerase 3 is really important for us because it elongates DNA. Okay, so it works in the 5' to 3' direction. It also is a very cool protein. It has a little bit of an ability to read in the 3' to 5' direction when it's synthesizing genetic material. So it has a little bit of editing capability as well. But a lot of the DNA repair and translation is actually going to be done by DNA polymerase 1 at the very end of this process. That's going to come through and remove the primers and replace them with DNA material. All right, so still talking about DNA polymerase. DNA polymerase 3 is the one that's going to be very important in the replication process, and it's the one that is actually utilized first because it is going to add nucleotides to the 3' OHN of the newly synthesized strand. And then DNA polymerase 1 is going to come in at the end of the DNA, come in at the end of the process and remove the RNA primer and replace that with DNA as well. All right, so we are going to end this section here, and then we will jump into talking about the rest of DNA polymerase's function and the rest of the replication process as well.